Okay, so sort of the plan for this evening and for this week is as follows. Tonight, I want to get through lectures 14 through 16, sort of some of the key points of those lectures. And then I also would like to sort of loop back and do mutations, which were affiliated with lecture 13, and I wanted to get to them last week, but ended up running out of time due to just how much info was packed into last week with transcription, RNA processing, and translation, protein synthesis. That was a lot, so I wanted to take my time there. So we didn't get to mutations, so I just have a little mutation question that we can do at the end. Yeah, and then, so that's tonight. Tomorrow night, my Monday session is also at 6 p.m. And it typically is in Tool 303 up on the 3rd floor. I'm expecting that we will probably have more people than usual tomorrow night, given that there's a midterm this week. So there's a good chance we might be moving down to Tool 3, the big auditorium downstairs. And then my Wednesday session is from 7 to 8. Also a good chance that we could be moving to the auditorium downstairs, depending on whether or not the auditorium is booked for that time slot. But my Monday and Wednesday sessions will be review for the midterm. So it could be anything from lectures 8 through 16. So that would be enzymes, nucleic acids, DNA replication, transcription, RNA processing, translation, and then these 3 lectures on gene regulation. So with that, I guess we'll dive right into gene regulation. If you have any questions, feel free to interrupt, turn on your mics and ask, send a question in the chat, whatever. Also, a lot of people have been asking me about recordings of my online sessions. They should be posted sometime today, I think, is what the email said. So sometime this evening. So hopefully that includes this session as well. I think it will. So yay. They'll be posted on the PAL D2L page. And I will also post all of my blank worksheets from the beginning of the semester until now. I'll do that after my session tonight if I think of it. But hopefully I'll remember. Okay, so getting into tonight's content, when are genes turned on? This is a really kind of vague question, but what I wanted to get at is that genes are only turned on when the protein or the mRNA that they encode is needed. So a gene will never just get turned on if there's no need for it. It's not like at every moment, every gene in your body is like ready to ramp things into gear. Most of the time, they're just sort of like chilling, vibing, waiting to be needed. For example, we talked about the tryptophan system. And really that, well, I guess we'll get into that. But, okay, yeah, sorry. We'll get there. But I just don't want to get into that whole thing quite yet, but we will talk about it. So yeah, genes are only turned on when the protein that they encode is actually needed. And I think, what does turning a gene on mean? That its protein starts getting made. Yeah, that's exactly what I mean by that. So basically, if you have a gene that encodes for a certain protein, if you have like a whole bunch of that protein already, then you don't really need to make it. So that gene is not going to be turned on until you run out of that protein and you need to make more. That's a really good question. And at what levels can gene expression be controlled? We talked about sort of 3 levels. So at the level of the gene, which is the DNA. We can have chromatin packing and transcription initiation. So basically what that refers to is, oh my goodness, there we go. Chromatin packing, we talked a while ago about heterochromatin and euchromatin. And if the chromatin is packed really, really tightly, then you can't really express that gene because there's nowhere for transcription factors to bind. 1 second. Okay, Teams is telling me I have a notification, but I don't think I do. I think it's just from the question that I already answered. Never mind. I do in fact have a Teams notification. Okay, sorry for this distraction. Someone was unable to find a link to join the meeting in their email, so they just asked if I could send it to them. So I'm going to try to do that real quick, and then we'll get back to this. Okay, hopefully that works for them. Okay, so yeah, at the level of the gene, expression can be controlled by the chromatin can be so tightly packed together that transcription factors can't bind, and so you can't really express that gene that way. And also transcription initiation is controlled at the level of the gene. Essentially, transcription starts with DNA, and if for whatever reason there's some sort of error in the initiation of transcription, then that gene is not going to be expressed and that protein is not going to be made. Okay, perfect. I think we're good. And gene expression can also be controlled at the level of, oh my goodness, the typos tonight, at the level of RNA. So depending on how stable the RNA product that you make in transcription is, RNA processing is a way of controlling gene expression. So we talked about an aspect of RNA processing is alternative RNA splicing. So if you are trying to make one certain protein, you might splice out a select group of introns, but if you take that same gene and you want to make a different protein with it, you would have to splice out different introns to make a different protein. So that is also an example of controlling gene expression. And also translation, initiation, and termination. Basically, translation entirely relies on your RNA strand. So if there are any changes in the RNA, then that will result in a change in your translation process. Okay. So then we also talked about controlling gene regulation at the level of the peptide, or the polypeptide, I suppose, which basically can lead to protein modification, ability and activity. I guess depending on what the goal, I guess, of the protein that you're trying to make is, or let me rephrase that, sorry. Gene expression can be controlled at the level of the polypeptide, which is like your RNA, or not RNA, sorry, your protein synthesis product, your translation product, via modifying your protein in some way. If that makes sense. I'm not sure that that made as much sense as I was hoping it would. But if that was gibberish, let me know. Also, I have just realized that my laptop is a little bit low on battery, so we'll see. I might need to take my charger out at some point, but it's right beside me, so we should be good. But yeah, if I won't let it get to the point where I just dip off the Teams chat by mistake. Don't worry. Okay, so that was just sort of like an intro. I wouldn't really worry about that stuff too much. But what you do need to know is what is a transcription factor, what is a repressor, what is an activator, that sort of thing. So transcription factors are regulatory proteins. They're proteins that regulate, essentially, and they bind to a specific DNA sequence, which is a gene, and they control transcription initiation. Oh my goodness, there we go. So, yeah, transcription factors are sort of, I mean, you can piece together the word, factors that control transcription, but specifically they are proteins they have like a wide variety of different functions, but they essentially regulate transcription, and they bind to the gene that is attempting to be transcribed, and they control that initiation of transcription, so the very beginning of it. Repressor proteins are another type of regulatory protein. Oh my goodness. There we go. And basically what they do is they turn genes on by enhancing the binding of RNA pull. So we talked about when transcription begins, it starts at the promoter, and the repressor proteins are these little proteins that turn the genes on by enhancing the binding of RNA pull to the promoter. So we know that RNA polymerase binds to the promoter, and then it starts chugging along and making an RNA transcript of the DNA. And basically it just, you can think of it as it enhances, well, that's exactly what I said, it enhances the binding. So you can think of it as sort of, it just makes a tighter bond and just sort of improves that interaction. Activator proteins are another group of regulatory proteins. Oh my goodness. I have given you the definition for activator proteins for repressor proteins. Okay, that makes a lot more sense in my head. I was like, that seems backwards to me. What? That's because it is backwards, because I, for some reason, thought that I had activator proteins first, but I in fact had repressor proteins first. So ignore what I have just said, because that is actually the definition of activator proteins, which makes a lot more sense because they are activating, whereas repressor proteins would repress. Okay, yeah, sorry about that. I was like, oh, like those aren't repressor proteins. And then I was like, oh, wait, no, I'm talking about activator proteins, but I was not. Okay, wow, that was a big oof. Sorry about that. So yes, activator proteins are those that turn the genes on, which makes sense because they are activating the gene. Repressor proteins repress the gene. So they are a different type of regulatory protein. And these turn the gene off, preventing RNA polymerase from binding. There we go. Oh my goodness, that makes a lot more sense in my head now. I don't know how I got those flipped around. My apologies. Okay. So yeah, the repressor proteins are the ones that turn the gene off by preventing RNA pull from binding. So RNA polymerase can't bind, so it can't chug along and make those, make the transcripts, the RNA version of the gene that you're trying to make a protein with. Okay, big blunder. Sorry about that. So the next thing that I wanted to just have a quick chat about was contrasting the transcription factor binding sites in eukaryotes versus prokaryotes. So in eukaryotes, the binding sites for transcription factors can be found super far away from the promoter. So they may be found thousands even of base pairs, or we'll say nucleotides, I guess. That's probably a little bit more accurate. So thousands of nucleotides away from their promoters. Typically, binding sites sort of cluster together. And yeah, eukaryotes are always, when you're comparing eukaryotes versus prokaryotes, eukaryotes are always a bit more complicated. We didn't really talk about gene expression in eukaryotes a whole lot, just that short little sort of half lecture on them on Wednesday. But yeah, I wouldn't worry about eukaryotes too much. The main focus that you want to have is prokaryotes. I would know a little bit about eukaryotes as well, but most of what you should know about this is going to be based on prokaryotes. In prokaryotes, transcription factor binding sites are often found close to the promoter and are called operators. So that is a good word to know. And an example that we talked about is the tryptophan repressor, and this sort of trip operon example. And so, yeah, I remember this being like really confusing and frightening when I took this course. So I want to like take time with it, and also like, yeah, we've got lots of time tonight. Yeah, okay, never mind, we're good. For some reason I was thinking I had been talking for longer than I have. But essentially, this tryptophan repressor, this whole system, is a good example of gene expression in prokaryotes. So basically, there's two scenarios with this trip operon. And those two scenarios are that you can either have tryptophan available or tryptophan is not available. And when tryptophan is not available, tryptophan in this sort of operon acts as a co-repressor. When you don't have any tryptophan, you don't have a co-repressor. And if you don't have your co-repressor, then your repressor, so your repressor proteins is what I'm talking about here, cannot bind to the operator. And essentially, in this sort of system that requires a co-repressor, tryptophan's the co-repressor, and that repressor needs its co-repressor enabled to do its job. So the repressor proteins we said earlier, once I got that sort of straightened out, we said that they turn genes off by preventing RNA polymerase from binding. But because the repressor doesn't have its co-repressor, then RNA polymerase binds. And the gene is turned on. Oops. And so this makes sense because when tryptophan is not available, it's basically telling the cell to make more tryptophan. So this is I'm trying to like find the right words to describe this. The wording can get very like sort of tricky. So bear with me, sorry. But when TRP is not available, then ideally the cell wants to make more tryptophan. So when TRP is not there, it leads to the gene to make more tryptophan being turned on. So this system is really great in that sense, because when TRP is not available, then it leads to the gene that codes to make tryptophan being turned on, and more tryptophan is made, and then everyone is happy. And then that leads to our second scenario where tryptophan is available, because ideally tryptophan was not available, and then we made more tryptophan, and now it is. And so what happens in this operon when tryptophan is available is that the co-repressor, which is tryptophan, bear with me, binds to the repressor. And now the repressor has its co-repressor, so the repressor can bind to the operator. And because the repressor binds to the operator, RNA polymerase cannot bind. Therefore, the gene is turned off. So when you have tryptophan in the cell, then the gene to make more tryptophan is turned off, which is ideal, because you don't want to make a whole ton of tryptophan if you already have some available in the cell. So basically, when tryptophan is present, it tells the cell, okay, we're here, stop making more tryptophan, like we got it covered. And then when tryptophan runs out, the cell is able to recognize, oh, we got to turn this gene on and make some more. So that's a really, really interesting system. And this is a repressible system example. So this example of the tryptophan is, I'm actually just gonna go like this so that it's all on one page, is an example of a repressible operon. And I see I have a notification in the chat. Does the co-repressor leave the repressor after it's changed the repressor's shape? That is an excellent question. Let me think about that for a second. So the co-repressor binds to the repressor, changes the repressor's shape so that it's able to bind to RNA polymerase and repress that reaction. Does it leave the repressor? I don't think so, but I could be wrong. I'm just trying to think, and I don't think there would be a reason for it to leave the repressor after it has changed its shape. But let me maybe just check, go back to the slides from this lecture and sort of look at like the visuals that were there. And just let me check that. That's a really good question. I've never really thought about that before. Oh, I'm looking at the lac operon. I'm in the wrong lecture, I think. Yeah. Okay. So I'm looking right now at like the visuals that are on Dr. Patton's slides, and I don't see that the co-repressor is leaving. So I don't think there would be any reason for it to unattach. But I do not know for sure. So yeah, I don't know. That is a good question. And I'm sorry that I don't have like a definite answer for you. You could definitely ask. Oh, but Dr. Patton is like gone now. Well, she'll be back Wednesday. So that could definitely be a question for Wednesday's review session. That's a good one. Yeah, sorry that I I don't know for sure. I would think that it would stay attached to the repressor. Just sort of to ensure that the repressor doesn't revert back to its original shape. But I just can't say with 100% certainty. So yeah, that's a good question. Sorry. Okay, yeah. So this is an example of a repressible operon where in the presence of tryptophan represses the system from making any more tryptophan. The lac operon is a tad more confusing generally. And there are a few more scenarios with this this operon example, because there is this activator protein that also plays a role in how high the gene expression is going to be. So in the sort of simplest case of this example, the lac operon, is when we don't have any lactose present in the cell. And basically, we have this lac-I here, which is a lactose repressor. And when there's no lactose in the cell, that allows the lac repressor to be bound to the operator. And when the lac repressor is bound to the operator, RNA polymerase cannot bind. And so that means the gene is turned off. So here's my sort of junky little illustration here. We have the promoter, the operator. This is lac-I, the lac repressor. This is RNA polymerase. It's trying to bind to the operator, but the lac repressor is in the way, so it simply cannot bind. When lactose is present, lactose will bind to the lac repressor and change the shape of the lac repressor so that it can no longer bind to the operator. So when lactose is present, lac repressor is not bound to the operator because lactose binds to the repressor, changes its shape, it's no longer able to bind to the operator. And so in this case, RNA polymerase can bind, and the gene is going to be turned on. So RNA polymerase can bind, and it can sort of chug along, and transcription can occur. However, there's sort of this extra factor to consider with the lac operon example, and that is glucose levels, because glucose levels are inversely related to CAMP levels. So CAMP, or cyclic AMP, is what is it actually technically called? Oh my goodness, I'm completely blanking. What is like the word for what CAMP is? It's not an activator protein. It is, oh goodness, what is it? Let me see if I can find it in my notes here real quick. Okay, I think the slides just refer to CAMP as a small molecule. I can't think of what CAMP actually technically is, but basically it is a small molecule that when it is present, it can bind to this activator protein called CAP, and sort of allow for CAP, this activator protein, to do its job. But CAMP is, oh wait, there's a notification in the chat, a messenger molecule. Yeah, I've definitely heard it referred to as a messenger molecule before. Actually, that is, I think, probably the sort of term I was looking for. Thank you. Yeah, I think that is exactly what I was looking for. Oh my goodness. Sorry that I seem like a little bit all over the place tonight. To be quite honest with you, I guess it is because my brain's a little all over the place tonight. But yeah, CAMP is this little tiny molecule that if you look in this diagram down here, it's this little black circle here. And essentially when CAMP is around, it allows for CAP, this activator protein, to sort of do its job. But CAMP levels are inversely related to glucose levels. So when glucose is around, CAMP will not be around. So in this example, cyclic AMP or CAMP levels are low. That is because glucose is present. And so without CAMP, the little messenger molecule, CAP, the activator protein, bind. So gene expression is low. Gene expression is going to be on, or the gene is turned on, but the expression is going to be low. So RNA polymerase is going to chug along, but it's not going to chug along very sort of quickly or effectively, is how you can sort of think of that situation. Whereas when we have this sort of third scenario where lactose is present and glucose is absent, the beginning of the situation is the exact same in that the lac repressor not going to be bound to the operator because lactose is present, so it binds to lac-I. Lac-I can no longer bind to the operator, so it's just going to be floating free. This means that RNA polymerase can bind. Oh my goodness, how many times am I going to spell polymerase wrong tonight? I always type it wrong. So the gene is turned on. This time, oh, come on, you. Hello? Honestly, I'm such a boomer. Okay, so this time glucose is absent. So when glucose is absent, CAMP levels are going to be high. So just like here, glucose levels were high, so CAMP levels were low. This time glucose levels are low, so CAMP levels are high. Hello. CAMP levels are high because glucose is absent. And so in this example, CAP, which is the activator protein, oh my goodness, can bind to RNA polymerase. So gene expression is going to be high. Let me try to move this again. Oh, no, that's not what I wanted. I wanted to move the whole thing as one. Come on. There we go. So gene expression is going to be high because this activator protein CAP is able to bond or bind to RNA polymerase thanks to the molecule, the messenger molecule, CAMP or cyclic AMP. So that is sort of like an overview of the three different scenarios that can occur with the lac operon. And the lac operon is an example of an inducible operon. And why that is, or how I sort of keep that straight in my head, is that the presence of lactose in the lac operon induces the system. So when lactose is present, the gene is turned on. When lactose is not present, the gene is turned off. Whereas back here, the tryptophan example, this is a repressible operon. So in a repressible operon, tryptophan, when it was present, repressed the gene and turned it off. Whereas in an inducible operon, the presence of lactose induces the gene and turns the gene on. And we get various levels of gene expression due to this activator protein cap. So when the gene expression is high, basically you can think of it as RNA pull is cranking along. It is whipping through this DNA, or I guess this transcription, transcribing RNA. And it's very effective. It's doing a really good job, is how you can sort of think of that. So, that leaves us time to go over mutations, which if you think I went a little bit too fast through this and you want me to touch base on it again Monday or Wednesday, let me know. I just wanted to make sure I left time to get through mutations. So feel free to let me know if you want to see this again Monday or Wednesday. But I'm going to switch over to mutations, which is here. Okay, so if you were at my Monday or Wednesday sessions last week, we talked about all of this good stuff. What's transcription, some sort of practice problems with transcription, what's RNA processing, and then what is translation? What does that look like? The three sort of steps. I recommended Mr. W and his music videos and some various YouTube videos on transcription and translation. Honestly, I recommend YouTube for almost every aspect of this course, but especially like DNA replication, transcription, and translation. There are just some really, really great animated videos out there that I find particularly helpful. Personally, I think that's just sort of the way I learn, is I really like to see things moving and not just, you know, like what I just showed you with the lac operon, sort of like boxes on a screen that are just like 2D and don't really move. I just find that like when it's in 3D and things are moving, it just really clicks with my brain. So I highly recommend that. Okay, but mutations. This goes along with translation. Mutations are not always a bad thing. They're generally regarded as being this negative thing, but they're not always. They get this bad rep because typically, like if you want to make a certain product and then there's a mutation and you end up with a different product, that's typically not good, but sometimes it can work out for the better. So just sort of remember that. This little table is important. Make sure you're familiar with this table and you know how to read it, you know how to use it. That'll definitely come up. I would be floored if it didn't, but I've been floored before on things that did or did not come up that I was expecting or not expecting. So, but the question that is being asked is, what type of mutation is each of the following an example of, and what is the effect on the resulting amino acid chain? So we're given this original strand of RNA. So this is our RNA transcript. But let's say that there was some sort of mutation in transcription and we ended up with different variations of that. So if we're looking at our original strand, we read strands in sets of 3. So codons of 3. So we would read as, also, disclaimer, I should throw out that you wouldn't ever typically sort of start just reading at a 5' end if you were asked to take an RNA strand and go, this is our RNA transcript, what is the resulting amino acid chain? You would look for AUG, which is your start codon, and you would start there. But this is just a random example. So we're not technically like trying to make a whole chain here. Just pretend this is like a random snippet that I took from in the middle of a piece of RNA. Don't think about it too deeply. Don't come for me because AUG is not at the start. I just wanted to like just, you know, use this as an example. So let's pretend we had an AUG before this, just for simplicity's sake. So we have AAG. We'll make her red, followed by UCA. We'll make UCA green, and then we have GUA, and I'll make her blue. So those are our 3 codons. And so we would go to our codon chart, and how you use those is you go to the first base in the codon. So looking here, it would be A. Second base in the codon is also A, so you know we're going to be looking in this block here where the 2 A's meet. And our third codon is G. So that lines up to be lysine. So that codes for lysine, followed by UCA. So we have U, C, and I guess no matter what our third base here was, but ours is A, UCA codes for serine. See a notification in the chat. Is the template not made in the 3' to 5' direction? The template, okay, the template is the DNA strand that you are working off of. So you're never going to be making a template. Your template is the DNA strand that you sort of start with, and you make an RNA strand from that piece of DNA. And the RNA that you're going to be making is always made in the 5' to 3' direction. So you can sort of think of your template running 3' to 5', I guess, because your template is going to be sort of anti-parallel to your RNA that you're making, and your RNA is going to be built 5 to 3. So like you can sort of think of your template of DNA running 3' to 5', but nothing, like no DNA or RNA is ever made 3' to 5'. It's always 5' to 3'. But good question. Oh, whoa, I just like pushed my desk by mistake. I don't know how that happened. I leaned forward with too much force. Oh, I should maybe color code this so it makes it a little bit more clear. So AAG gives us lysine, UCA gives us serine, and then GUA would give us, we would go G, we would go U, we would go A, gives us valine or valine. So this would be the amino acid strand that we would make from this strand of RNA. One sec. Okay, so in the chat, so we read the original strand from 3' to 5'. Can we say, starting from the right side, AUG would be the first codon? AUG is always the start codon, yeah. And you're gonna be, I guess I'm not sure what you mean by the original strand. So do you mean like the original strand of DNA? Or like the strand of mRNA that you make your protein with? Okay. Oh yeah. Okay. That was silly. Yeah, the original strand. I literally have this labeled as original strand, so that was a silly question of me. Okay, so we read the original strand from 3' to 5'. Can we say, starting from the right side, AUG would be the first codon? When the ribosome reads the RNA strand, it is going to be reading 5' to 3'. So you would start from the 5' end, which in this case is on the left. So you would start here. And yeah, maybe I just made things more confusing by saying, oh, like this isn't the start codon, like bear with me, but maybe, okay, maybe just for like confusion's sake, or for lack of confusion's sake, I'll throw AUG in there, and let's say that there was an AUG right here. That codes for methionine, and then we would have lysine, serine, valine. Yeah, so your RNA strand is also going to be read by the ribosome 5' to 3'. So when I was saying that the template, you can sort of think of it as being 3' to 5', that was more referring to when you're like transcription. So this is sort of getting a little bit unrelated, going all the way back to transcription. This is your piece of DNA that you're looking to transcribe. And when you're trying to figure out which is the template strand, it says right here, the top is the template strand. And a lot of people, when we were talking about figuring that out last week, found it easier to think of, well, your promoter is here, your gene is here, so you're looking to build from right to left, and you have to build 5' to 3'. So you can think of your template strand of DNA as running 3' to 5'. So that is sort of like what a lot of people like used to sort of remember which is going to be your template strand, but you never, ever, ever, ever make DNA 3' to 5' or RNA 3' to 5'. It's just not possible to build it in that direction. You always have to build it 5' to 3'. So this way or this way. I hope that makes sense and is not like confusing you further. It's so like difficult to sort of communicate via Teams. So my apologies if I've been a little bit vague. Or if you're able to come to either of my sessions in person this week and you want to have a chat, feel free. We can continue that conversation there as well. Or if I still have not answered your question and you want to like give it one more shot tonight, I'm open to attempting that as well. So yeah, hopefully that makes sense. Okay, so getting back down into this example, I'm just going to throw AUGs at the start of all of these. And then the question is going to basically remain the same. So all of these examples represent a different type of mutation. So here we still have AUG. That's fine. That's our start codon. So our ribosome is going to start reading there. Then we have AAG, which is, that is what we had earlier, AAG. So everything's good. We'll color that red. But now we have CAG is our next codon, and so that is different from what we initially had. So we'll color that orange maybe. And then we have UA, which isn't even a full amino acid. So what has happened, or it's not a full codon, sorry. What has happened here, our AAG is still the same, but then we had a mix up here. So we went from UCA to CAG. And what happened is that this U went missing. So there was previously a U here. We would have had UCA and then GUA, but the U is missing. And that is known as a deletion mutation. Sorry, excuse me. And so what this is going to give us is methionine, and it'll give us lysine, which is what we want. But then CAG is going to code for A, G. Oh goodness, is that GLN? This is a little bit blurry to me. I need to get my vision checked. GLN, what amino acid is that? Is that, oh, whoops. Oh my goodness, glycine, I think? I think it's glycine, but I'm pretty sure. Yeah, and so that obviously is going to have a big impact on the resulting amino acid chain because we don't have serine or valine and we have this new random amino acid that we didn't really want. So then in this example, we have AUG, which is methionine, so that's all good. And then we have AAG, which is all good. That's what we want. That's going to give us our serine. And then we have CUC, which is not what we want. This is going to be something random. Let's color that one purple, just for no real reason. So CUC and then AGU. And we'll color that one. Running out of colors. We'll color it this nice, pretty blue. So this is going to give us methionine, lysine from the AAG. CUC is going to give us leucine, and then AGU is going to give us serine. So lysine is good. That's what we want, methionine followed by lysine. But then leucine and serine are just not what we are looking for. And so again, our amino acid chain is going to be different from what we originally wanted it to be. And what happened here, what the mutation is if you look, we have AUG, AAG, that's all good. But then here we have a C that wasn't there before. Should just be UCA, GUA, but we have this random C that was inserted, and that's called an insertion mutation. And so deletion and insertion mutations are both called, oops, probably put it in dark ink. These are both frame shift mutations because they shift the reading frame. Because we read in groups of 3, AUG, AAG, you know, or I guess this is a better example, AUG, AAG, UCA, GUA. And by adding in or by taking away just one or adding in just one, that messes everything up in terms of how you read. And so a good example about like reading frames is, you know, the sentence, the cat ate the rat. But if you take away this H, we have a deletion mutation. That would then read TEC, ATA, and then TET, HER, AT. Tech, atta, tet, her, at. You know, and you can see that comes from TEC, ATA. Oops, I didn't mean for that C to get highlighted. ATA, okay, that C just wants to be highlighted. TET, HER, AT. So yeah, that's just sort of an example of why frame shift mutations are so yucky and disgusting, because when you read in groups of 3 and only one thing gets taken away or added, it just messes everything up. So those are frame shift mutations. And then we have, oh, there's not enough room for that, but we'll just move down here. We have this other version here, which is AUG. And then we have AAA, which is different from what we've seen before. Like I said, I'm running out of colors. We'll make it this weird mustardy color. And then we have UCA, which we have up here. That's our serine. And we have GUA again, which is our valine, which is also what we want. And so the issue here, it's not a frame shift mutation, this example. What's happened is that this G has simply been replaced with an A. And so this is called a substitution mutation. And so this is going to give us methionine. AAA codes for AAA. Also, just so happens to code for lysine. UCA codes for serine, and GUA codes for valine. So we're actually going to just coincidentally end up getting the exact same chain. So this is called a silent mutation. Typically, substitution mutations are the least sort of dangerous mutations because they don't interrupt the reading frame. They, oh, sorry, I was just checking my laptop battery. They, in this specific example, by replacing that G with an A, if you look at AA, and then you get to this box, AAA and AAG both code for lysine. So that substitution made no difference on the amino acid chain. If the G would have been substituted for a C, then we would have had asparagine instead. So that wouldn't have been ideal. We would have had methine, lysine, asparagine like that, but serine and valine would have stayed the same. So it would have just been one amino acid that ended up being wrong instead of all of the amino acids that came afterwards being wrong. So substitution mutations are typically the least impactful, you could say. And often they end up being silent mutations because the genetic code allows for that to happen. So we are at 6:57. So I don't really think we have much time to sort of start anything new this evening. But if you have any requests for things that you want to see covered on or tomorrow or on Wednesday, please let me know. Shoot me an email, send me a message on Teams, or you can just bring your questions tomorrow night. I will be there. Hope to see you there too. Thanks for showing up tonight. I know Sunday night you probably have maybe slightly better things to do on your Sunday night, but I always like having lots of people at my PAL session, so I appreciate it. If you have any questions, feel